Here's an example of this. So this is a cuvette of fluorescent dye where the objective on the right is focusing infrared light, and the objective on the left is focusing ultraviolet light. And from the objective on the left, down at the bottom, you see this sort of double cone-shaped cylindrical-like structure where you excite fluorescent dye throughout the path of the ultraviolet light. On the other hand, from the infrared light, you can see that there's just one small three-dimensionally localized volume where the fluorescence is excited. So now the idea to build an image is to excite these fluorescent molecules and then to scan the position of the laser focus around inside the material and record how much fluorescence you get as a function of position. One thing I didn't mention is that this tool, this technology actually requires very short laser pulses, 100 femtosecond roughly duration laser pulses. And the reason for that is just the probability of driving this nonlinear optical process is quite low. So in order to drive it with any kind of efficiency with reasonable average powers that won't damage the sample due to residual linear absorption, you need to pack the light that you have into a series of short pulses that are separated in time. Okay, so I just want to show you an example of nonlinear microscopy. So this is a three-dimensional image stack where we're scanning down into the brain of a live anesthetized rodent. In this case, the blood plasma has been injected with labeled with a fluorescent dye, so you can actually see all of the blood vessels inside the brain. And and this is going down about 1 millimeter into the brain. And to play it again, and when we start, you'll see some of these big surface vessels up on the top, and then those plunge down into the brain and give rise to capillaries. And the little black dots that you see zipping around inside the capillaries, those are actually individual red blood cells. So it turns out the dye that's injected to label the blood plasma doesn't label the red blood cells. So they show up as dark patches. We actually track their motion in order to extract blood flow speeds. This kind of imaging has enormous power. Here's just an example of some two-photon imaging from my lab. Blood vessels are labeled with a red fluorescent dye. And in this case, this is an animal that expresses a fluorescent protein, yellow fluorescent protein, in a subset of the neurons in the brain. And the pair of images at the top are images over a depth, projected over a depth of the top 100 microns. And you can see individual dendrites in green and big blood vessels in red up on the surface of the brain. The pair of images below are actually projected also over 100 micron depth, but they're between 600 and 700 microns beneath the surface of the brain. And there you can see that the imaging quality is actually not degraded at all. We have the same contrast, same resolution as we had up at the surface, and there's actually not loss of image quality, at least in our hands, until a depth of about 1 millimeter. And so in those lower images, you can see finer blood vessels, capillaries, and the cell bodies in green of those pyramidal neurons. There's other nonlinear optical methods that have been used for imaging. For example, harmonic generation. So this is an example of third harmonic generation where 3 infrared photons are coherently converted to 1 ultraviolet photon. It turns out this is a very interface-sensitive microscopy because of the Gouy phase shift that light undergoes when passing through a focus leads to cancellation of light unless there's something that breaks the symmetry of the focus. And on the right, this is actually a cross-section of the spinal cord of a mouse. And the things that show up a little bit brighter are actually the myelinated axons in the spinal cord. And they show up brighter because myelin is essentially a whole bunch of stacked interfaces. And those interfaces break the symmetry of the focus and lead to more third harmonic generation. Coherent antistokes Raman scattering microscopy developed in Sunny Shi's lab at Harvard makes use of Raman transitions. In this case, 2 laser pulses are sent into the sample simultaneously, a pump and a Stokes pulse. And through what's essentially a resonantly enhanced four-wave mixing process. You can see the energy diagram on the upper left. You generate light at a blue shifted frequency, the antistokes frequency. And whether or not the process is resonantly enhanced depends on that, the difference in frequency between the pump and the Stokes beam matching a vibrational resonance, matching a Raman active vibrational resonance in the molecule. And this has turned out to be a fantastic way to image lipid. In this case, most people focus on the vibration of a hydrogen atom on a carbon, and lipid has lots of carbon-hydrogen bonds. And it turns out that lipid is something that's difficult to fluorescently label, at least in vivo. There's no fluorescent labels that you can use to look at lipid that don't also disrupt the lipid structure. So these are some images of lipid that's been taken up by live cells. So this is what happens when you eat a really fatty hamburger. And over on the right is actually some two-photon microscopy that shows that these lipids are being incorporated into lysosomes and targeted for degradation. One thing sort of interesting here, the image on the lower left is actually taken at a modestly different frequency, and the molecule that's been imaging has been deuterated. So the hydrogen atoms have been substituted for deuteriums, which are heavier, and so they change the vibration frequency of the C, now D stretch. And this gives a way to distinguish a couple of different lipids from each other. Okay, so all of these methods, the nonlinear effect that was being looked at was measured by looking at a large change in color. So we sent in light of one color, and we looked at how much light comes out of another color. So we focused short pulses into the sample. There's a nonlinear interaction in the focal region that generates a new color of light, and then we map how much of this new color of light we get as a function of position in the sample by scanning the focus around. And that new light could be generated by a non-parametric process like two-photon excited fluorescence, or by parametric processes like harmonic generation and coherent antistokes Raman scattering. But what about all the other kinds of nonlinear effects that don't result in color changes? Couldn't we use those to image? So the overall idea would be the same. We map the degree of the nonlinear effect that occurs as a function of position inside the sample to build up an image. But now we would be looking at things like self-phase modulation and nonlinear absorption, excited state transitions or stimulated transitions, or things like degenerate four-wave mixing, not like the highly non-degenerate four-wave mixing of third harmonic generation. But now we have a problem. So one of the reasons that two-photon microscopy and these other things work so well is that there's no light being emitted at the color that you're detecting. You're only sending in infrared light, and you're only measuring, say, green light coming out. And there's no background green light, so you get to make a null measurement. You measure how much light there is away from a background of 0. But these other effects here, there is going to be a large background, and these are nonlinear optical effects, so they tend to be small. So you're going to have to measure a very small change on top of a large background. So how are you going to be able to do this with high sensitivity? And it turns out that people have been doing this for years in the pump probe spectroscopy world. And there's perhaps a lot of ideas and thoughts that could be mined to turn into imaging tools. And I'm going to show you just a couple of these today. So in terms of strategies for being able to see small changes on top of a large background induced by a nonlinear optical effect, I'm going to show you one strategy, often called modulation transfer, where the idea is you have a two-beam nonlinear optical interaction, and you modulate one of those beams on and off very rapidly, and you look at the effect on the other beam. So only when both beams were there will there be this nonlinear interaction, and that'll affect the intensity or the phase or some other aspect of the non-modulated beam, and you can detect this with high sensitivity using lock-in amplification.